What's up about payments? Today we're going to do FRQ Friday, and we're going to look at tips and tricks for the AP exam. So I'm going to talk about different strategies, and then I'm going to really deep dive into different types of graphs, and I'm going to talk about experimental design and how we can get some of those easy points for the FRQs. So first thing, I just want to make a public service announcement, y'all. This happens every single year where students get so into writing their responses that they accidentally write their response on the wrong page. So they're, going through question 3 and they're, writing, and then they flip the page and then they see at the very top of the screen, it says question 3, write your response to question 3 on this page. And then you sit there and go, oh my God, I wrote on the wrong page. And I see it every year. Students will rewrite their entire passage, their entire response in a new location in their book. So do me a favor, just take a deep breath. And on the page where you're supposed to write that FRQ, just tell them where to go. So see question number 2. So whenever the readers get a blank page, they will go full book and they'll look at all of the responses, but they more or less are kind of like skimming. And so this ensures that your reader can find your response to ensure that you're going to get your points for what you wrote. So it doesn't take that long. Just write C question 2, and then it helps the reader find your response, and you also don't have to rewrite their whole thing. So first thing, where can we practice FRQs? So AP Central is great. So if you go to AP Central, you can actually see all the past FRQ questions. So if you click here, you'll see the questions, you'll see the scoring guidelines. The Chief Reader Report gives kind of a little bit of background information on what students did well or what they didn't do well. So you can kind of see what the nation had misconceptions in. The scoring stats helps you to see kind of what the national average of the question is. So personally, whenever I'm having my students practice and they're making, let's say make a 3 on the practice question, and they're all feeling upset because it's a 10-point question. They only got 3 points, and then I show them that the nation got 2 points. It helps you feel a little bit better. So definitely check out the scoring statistics just to see kind of how do you compare to the nation. Your goal is always to do better than the nation. And then over here we have sample responses. So they go and they usually have about 3 different responses. There's one that had like a perfect score, one with like a mediocre score, and then one with kind of a lower score. And it helps you to really see where the points were obtained. Plus it helps you to see from a student standpoint how to answer these questions. I've gone through every single FRQ from 2013 to 2024, excluding 2020. And I've kind of explained them as well as I showed the exemplar response and showed you where students got those points. So I call those FRQ Fridays, which is what this, series is. So if you go to my website, you'll see those. I've grouped them based on the question number, so the years, and I've also grouped them based on the units. So whichever way you're needing to have practice, you have access to those videos. So I've seen a lot of students writing questions about the exam, and a lot of them keep asking me, how are they going to graph on the computer? So I want to make sure that you're all clear that, yes, we're doing Blue Book, but you will write your FRQs physically in a book. So this is a hybrid exam. So section 1, which is your multiple choice section, you will access your questions through Blue Book, and then you'll answer your questions through Blue Book. So you'll just click the answers just like you did with the PSAT or the SAT. Now your second section, which is the FRQ section, you're going to get your questions in Blue Book. This maintains test security, but then you're going to answer those responses in a physical book. It's like a book. If you go to AP Central, you can actually see what this book looks like. It was similar to the page I showed you a second ago right here, where it just has the question number at the top, and it just says, write your response. And it's just like a book of lines. And so you'll physically write your graph. You will physically write your responses. You will physically annotate or modify a diagram for number 5. So there will be a physical book in front of you. So in terms of timing for the exam, section 2 is 90 minutes. You'll have 2 long FRQs and 4 short FRQs. I probably should have put the points on here, but the 2 long FRQs are between 8 to 10 points, and together they'll make 18. So either it'll be first question's 8 points, second one is 10. They'll both be 9, or it'll be a 10-pointer and an 8-pointer. You can usually tell how many points it is based on the bolded term. So your bolded term is usually 1 point, unless we're talking about graphing. And usually graphing is 3 points. Your short FRQs are always going to be 4 points, and so since we have 4 short FRQs, that's going to be 16 points there. So that gives you a total of, 34 points in the FRQ section. So in terms of timing, give yourself between 20 to 25 minutes for your long FRQs, about 8 to 10 minutes for your short FRQs. So in terms of like timing for the exam, this is kind of the strategy that I've shared with my students. So I'm of course sharing it with you. Take about 8 minutes or so to do a reading period. It's not required, but I'm just giving you my suggestions. Go through and look through the different questions. Find the ones that you know well, like the ones that you feel really confident about, versus the ones that you don't feel as confident about. You want to answer the things that you know first, and then leave the ones that you might not know so much about for the end. So if you happen to run out of time, you're running out of time on things you weren't really sure about in the first place. So spend about 20 minutes and write your first long FRQ. Then spend 8 minutes and do short FRQ and then a short FRQ. And then do 20 minutes for a long one, and then again do the two short ones. So this ensures you're kind of getting all those points kind of around the kind of the range. And then that also gives you about 10 minutes at the end in case you ran over on any of those questions, or kind of lets you to fill in anything you might have to. Sometimes you get so into writing that you don't actually see what you wrote. And if you take that 10-minute moment, you can read back and say, wait a minute, I didn't mean to say that. I used the same word twice in the sentence, and you can kind of fix some of those errors. So it kind of helps you to catch some of those mistakes you might have made. So in terms of the format, the first two FRQs are all interpreting and evaluating results. The second FRQ is where you're going to see your graphing. So it is guaranteed the second question will be a graphing question. Your third question is scientific investigation. So they give you some type of experiment. You're probably going to do some experimental design. The fourth one is conceptual analysis. So they're going to, again, probably have some type of experiment, but you're pulling the biological concepts from it. 5, you're going to have a model or representation. This is also where we're going to modify a model. Like 1 year we had to put an X on a phylogenetic tree, an X on a cladogram. We had to add an invasive species to a food web. So you're just modifying some model that's out there. 6, we're analyzing data. This is where box and whisker has come up a couple times. So they're usually going to give you some type of graph, and you're going to analyze the data in that graph. So some best practices. When you're doing FRQs, answer them in knowledge order. So if you know part B, just go ahead and write B and then answer B. If you know D, write D and then answer D. Like, you do not have to answer them A, B, C, D. And also, since you're answering them in knowledge order, you don't have to leave space for the one that you didn't answer. Just write them as you go down the page. And you'll notice on the paper it says like don't skip lines. They mean don't skip lines in between every single line. So like if you have part A and you write it out, you can skip a line and then do part B. Like, you're able to skip lines in between your passages. So label those responses. It makes it easier for the reader. You're not required to label your responses, but it does make it easier for your reader to find it. And then it also ensures that you can get those points if they can easily find your responses. Use the diagrams. So there's diagrams, there's prompt, like there's so much information in these questions. Don't get overwhelmed by them. They did rewrite the kind of the format for it last year, in which you have like a little bit of prompt and then the questions and then a little bit more prompt and then the questions. So this does kind of help a little bit with that over anxiety of how long the prompts are. But definitely use those diagrams, use those prompts. Define your terms. So a lot of times you'll just drop a phrase like, oh, this was crossing over, and then you don't ever like demonstrate how you know that this was crossing over or what crossing over is. So be very careful about just name dropping. If you're going to say that this like you're describing some term like this thing, then you can describe, okay, well, this is crossing over. I know this because I see that these two non-sister chromatids, they exchange information. Like, you can, explain some of it more. You want to label your diagrams. So I was talking to my students this week about like Punnett squares, and they said, okay, well, can we use a Punnett square to explain our response? Well, yeah, but whenever you use some type of diagram that you've made yourself, just label that diagram and say like figure 1. And then when your response, and you can say as seen in figure 1, 50% are this phenotype or this genotype and 50% are this genotype. And so it helps the reader to kind of follow where you did their work. Also, the first response is scored, so clearly denote that response. So if they say like, name 2 ways that this is, modifying, say the first way, the second way, so that they know where your 2 that you're showing are. Because if you just start labeling them, if you start writing them out, sometimes you accidentally put 2 together into one sentence, and then that counts as your 2. And if one of them wasn't right, then you automatically don't get your second response. So you always want to attempt the easy questions. And when I say easy, I mean like experimental design questions are easy questions. Like, you should be able to tell us independent variable, dependent variable, a control, a null hypothesis. Those aren't designed as like hard questions. So those are like our easy questions. You should be able to do a graph. You should be able to at least put something on the diagram. Even if you didn't put the right answer, you need to at least attempt to answer these easy questions. And then also read the questions. So sometimes y'all get so into writing your response that you forget what you were supposed to be answering. So make sure that you ATP answer the prompt. So let's talk graphing for a sec. So we want to use pencils. So on your exam, you are able to use pen or pencil. I recommend that you use pen for the exam, but on the graph, let's use pencil. We want to make sure that we label our axes. So put the title and then the units. Oftentimes this is given to us in the chart. So they'll be giving us the data chart, and in that chart we have our title and we have our axes. Sorry, our units. And then we want to use some type of logical easy counting. Okay, so when you label your axes, make sure that you're not squishing your graph. So you can use as much of the graph as possible. You don't have to start at 0, 0. So if your data is like all in the hundreds and you have like 120, 130, 160, you don't want to have 0 to that and where you're counting by tens, because then all your data is squished at the very top of your graph. But if you start at the bottom and you say, okay, well, I've got 110 down here, and you make each one 5, that would allow you to have more space, and your reader can ensure that you're graphing the points correctly. If they can't ensure that those points are graphed correctly, they can't award you points for that. So don't squish your graphs. And as I said, you're not required to start at 0. So if you want to do 0, then you can break your axis and then jump to another number. Just make sure that if you break your axis, you don't have any data points below the break. But you also don't have to start at 0. You can start at like 110 or whatever number you want to. Watch if the question tells you the type of graph. There have been some questions in the past that have told you make a bar graph, make a line graph, and students don't see that part and then they make the wrong graph. If you're doing a bar graph, then and you have your error bars, make sure you don't like cover up the bottom of that error bar. And then of course, like make sure your bars are wide enough that they're easy to see. Like if you're doing a single, like a single square and you're just wiggled all over the place, it's hard to follow where those data points are going. So just make them kind of wide if possible, and then make sure your error bar is not as wide as your actual bar. So I have a bunch of questions put in so we can kind of look through different graphs. So this one says, using the template in the space provided, construct a appropriately labeled graph based on the data in table 1, determine, and then they ask us to do something, okay? So looking at this, we can kind of say, what do we think is happening? Well, I'm looking at individuals and I'm looking at their glucose levels, okay? They gave me the axis already labeled. As you see, they broke their axis, okay? So what do I, as a reader, the student, have to do? Well, I'm going to have to definitely label my axes. So I'm going to put my individuals along the bottom, and I'm going to put my average blood glucose level and then my units along my y-axis, okay? So we want to make sure we label our axes. And as I said, they're given to us in the chart. So we see individual, we see average blood glucose level, and then we also see our units. So make sure you, for the most part, you can probably write this entire thing on there and it ensures you get that point. So looking at this, they are categorical data. Individual 1 has no effect on individual 2. Like this is me just looking at the relationship between these two things. So this is going to be considered a bar graph, okay? So we'll label each of our bars, okay? And so you'll put your labeling along the bottom. This is individual 1, 2, 3, 4, yada, yada. And then you're going to graph the mean as that bar, okay? So as you see, we have 170. You got the 170. We've got the 190, the 190, okay? And so you just make that with, the top of the bar, that mean, okay? Now notice here that we have this plus or minus. That denotes that we have a error bar. So I'm going to add 15 to the 170, and I'm going to subtract 15 from the 170, and that gets me my error bar. So I'm going to go from, 185 to 55? For that data, and you would do that for each of yours, okay? So on on PowerPoint, I was able to make a single line to do it, but you are welcome to put the little lines on the top and the bottom to denote the top and the bottom, which is fine. You could just make the single bar, okay? So then I say, okay, well, we want to determine, actually, the points were given axis label, bar graph, and then error bars, okay? So they said, okay, well, determine one individual who is at risk for developing a disorder and then also has a different blood glucose level from IV1. So first thing is, how do we determine about if things are different? Well, things are statistically significant if they are going to not overlap. And so we would say that they are statistically significant and they are different from one another if these error bars are not overlapping with each other. So I'm going to draw a little double arrow or thing or line or whatever this is to say where the axis is, right? Where I'm looking for things that are at risk, right? So here are the at risk. They're 140 to 199. And then I want to say, okay, well, how many of them are different than IV1? So I'm going to do a little circle there, and this is how far IV1 goes, okay? So I can see that individual 2 overlaps with that error bar, and I can see that individual 4 also overlaps. If I look here at individual 3, it does not overlap with the error bar from individual 1. So that tells me that individual 3 is both in the range as well as it is statistically significant, and it's different from IV1. And so we would have seen IV3 as our answer there. Okay. So in this graph, we see that we need to again use the template to provide a response and construct a appropriately labeled graph to represent the data in table 1. So here I see that I have 3 different things. I've got the strains, I've got the double strand breaks, and the average number of crossovers. And they say I'm looking for a correlation between double strand breaks and crossovers, okay? So these are the things that will be on my axes. And they already labeled my axes for me just to make life easier, okay? So because of the fact that I'm looking at 3 different things, I'm looking at each of these individual strains, but I'm also looking at this independent dependent variable component, right? And how those are related to each other. So this is going to be a scatter plot instead of a bar graph. Okay. And so here would be our scatter plot. We would label each of those points individually. Okay. You would have your mean labeled, and then you would have your error bars coming out from it. And this is what I meant by having the lines on the top and the bottom. Okay. So they said, okay, we'll determine whether corn strains, I'm sorry, the points you would have gotten was scaling, accurate error bars, and then that you made it a scatter plot where you had XY graph with separate points. So you could not make this a bar graph. You couldn't make this a modified bar graph. It had to be a scatter. Anyways, so determine whether corn strains 1, 2, and 3 differ in average number of crossovers. Again, we talked about that if they're different, it means that their error bars are not overlapping. Okay, so if I look here at 1 and 2, I can see that their error bars do not overlap with each other. Okay, they're close, but they do not overlap. That's another reason why I make sure that we are using as much of our graph as possible. So here you can see that person, they broke their axis, but they probably could have used way more of their graph paper, and that would have ensured that they kind of had a little bit more space to be able to see those overlaps. Okay. Anyway, so 1 and 2 do not overlap, so we would say that those are statistically significant and that 1 is higher. If I compare 1 and 3, okay, I see that those error bars also do not overlap. If I compare 2 and 3, I do see that they do overlap. So you can say there's no difference between 2 and 3, but that 1 is statistically significant and it's higher than 2 and 3. So as I said, we can see that 1 and 2 error bars do not overlap, which is why we say they're different. 1 and 3, again, do not overlap, which shows that they are also different. But as you see here, 2 and 3 do overlap, so they're not different from each other, okay? So another graph you might see is a box and whisker. I do not know if we're going to have to graph this ourselves, but it's one of the given graphs that's on the CED saying as graphs that students might need to know. So I wanted to go over the graphing of a box and whisker. We haven't had to graph one. I feel like it's more of a statistics thing to have to graph one, and that y'all are probably needing to analyze it, but I don't really want to leave you without resources. So this is the question from 2023, number 6, and they gave us the box and whisker. So just in case you don't know what a box and whisker is, on the box and whisker, the way that it's labeled is that you've got your median is this line that's in the middle of your box, and then the middle 50% makes up the box of that box and whisker. And then the whiskers would be the lowest 25% of the data and the highest 25% of the data. So this is just showing you your spread of your data points. You can see 50% of the data that's in the middle, and then you can also see where the median point is. And then again, you've got the 25% on each of the extremes. So they say, okay, well, based on the data in the figure, identify the gene that has the lowest median CQ value, where the Bs are different developmental stages. So they tell us that we're looking at developmental stages, which would be this little area right here. And I'm looking for the lowest median. As we mentioned before, the median is that line that we have in the middle. So we're looking at where is the lowest one of my medians, the lowest part where I've got that little line circled. I was just trying to draw your eyes to it. So we can see that in this little shaded one right here, that is the RPS5. So RPS5 is where we're going to find the gene that has the lowest median value. So this is an identify point, so you would be able to just write the single word. Personally, I tell my students to still write this as a complete sentence. Then I wanted to, give you a little bit more information. So CQ value is inversely proportional to the amount of mRNA from that gene. Based on the data in the figure, identify the gene that has the lowest level of gene expression. So if it's inversely proportional, right, and gene expression is the making of the mRNA, then we're going to find that wherever we have the highest point in our graph, so the one that has the highest CQ would correspond with the lowest amount of mRNA. So this point right here where we have that highest, so that would be the TBPAF. So again, we're just looking at the graph in order to pull information out of it. This is an example of what you might see on number 6. So here I've got growth curves for species B in group 1 and species A in group 2. They've got the shaded columns. We want to plot that on the template. So looking at this, it tells us that we're going to complete a line graph. So make sure you are looking, but if we weren't told this is a line graph, how would I know? Well, here you can see that I've got time going from 0 to 70. It goes in increments of 10. So this is kind of like a linear scale showing you that we're going and looking at something over time. So I would use this as a line graph because of the fact that it's linear data versus in the previous examples of the bar graph being categorical and the scatter plot seem to be kind of more like independent variables or independent values. So we were able to see those. So here they gave us this, which kind of threw some students through a loop that some of the parts were already labeled. So you got to figure out what your axes are already labeled as. As you see, species B is kind of laid out, and they said that it was the what are we shading? It's the unshaded columns. So we wanted to look at where it was leveling out for species B. So that means that this must be the species B grown separately. And so this right here would be 700. And then you would graph it from there. Okay, so we put our time on the X, we put the number of individuals on our Y, and then we of course label everything. As I said, this is our 700 because of the fact that species B kind of levels out. So we were able to get that from the data. And then once I figured out that that was 700, I could then kind of figure out, okay, well, species A got to 1400. So this is 1400 and I just filled in the middle, right? And so you would just label those points. I guess I didn't actually label, like graph the other parts for you, or when I transferred it over, I must have missed it. But you would have had another graph up here and one down here. So let's talk about experimental design, hot topic. So experimental design is definitely going to be on the exam. There's really no way for us to prepare you for what experiment you're going to get. But what we can prepare you for is what kinds of questions they can ask about that experiment. So first thing is you want to be specific about the experiment that's been given in the prompt. So they ask you about an independent variable or a dependent variable or a control or a null hypothesis. You want to be specific about that experiment when you're stating what that independent or dependent or control is. Okay. So the independent variable is what the researcher is modifying. So in the prompt, they're going to say, the researcher changed this. The researcher tested different blobs. Like they're going to give you something in the prompt that tells you what the researcher did. Then they're going to talk about what the researcher measured. The researcher found this data. The researcher collected this data. The researcher measured this data. So you're seeing what did the researcher get out of it, because the dependent variable changes based on that independent variable. So here we see we have to identify the variable in the experiment. I, of course, deleted the whole prompt from you, and I'm just giving you data table because sometimes you don't have to actually look at the full prompt. Okay, so if we look here, we see that I've got GTP either there or not, and I see cholera is there or not, and I look at the rate of CATP or cyclic AMP, sorry, production. So I can see that my independent variable is whether the GTP and the cholera is there or not. So I'm just looking at the presence or absence of the cholera or the presence or absence of GTP. On this one, you did have to mention presence or absence because in the chart it was either there or it wasn't there. So you did have to denote both presence and absence. And then say, okay, let's do this independent variable in this follow-up experiment. So they gave us a follow-up experiment. They're creating a modified version of CTP in which there's a DNA binding portion has been removed. I want to compare the frequency of crossing over in yeast cells in the presence and the absence of the CTF, which in the yeast cell is the presence or absence of the modified CTF. So we want to identify what the independent variable is. Well, in this case, since what are they changing? They're changing whether we've got that CTPF or whatever, CTF in there. Okay, so we're looking at the type of CTF. Was it the modified or unmodified? Or we're looking for the presence or the absence of the modified or unmodified CTF. So they're just, this is what they are changing in the experiment. So that would be our independent variable. So then here we're looking at fireflies. They emit light. in the reaction, they react to this other whole thing. They show you a figure. In order to determine the optimal temperature for this enzyme, scientists added ATP in a solution containing luciferase and other substances needed for the reaction. They then measured the amount of light emitted during the first 3 seconds of the reaction when it was carried out at different temperatures. So our dependent variable is what they're measuring, right? So it says they measure the amount of light. So our dependent variable is the amount of light. So here they say, based on table 1, identify a dependent variable in the researcher's experiment. So looking at the experiment, I can see that we've got this APase activity in high PI and low PI. I can also see that we have the different amounts of the RHO1 in the mRNA. So I can see that they're comparing the APase activity in the high and the low P, or they're comparing the relative PHO in the high and the low P. So you're looking at the APase activity as well as you're looking at the amount of PHO, because what they modified is they modified over here looking at these mutations. So a null hypothesis. So I found that students sometimes have issues with what the null hypothesis is. The null hypothesis is more or less stating that the treatment had no effect, that whatever we are doing is completely the same as the control. So I've kind of come up with a way that I've told my students where I say the independent variable has no effect on the dependent variable. Okay. And so I put independent variable and dependent variable into quotations because I don't want you to write independent and dependent variable in there. I want you to use the prompt to fill in those words. Okay. So here we have some sand lances. We feel like they're the keystone species. We want to see how that population affects based on the rising temperature and carbon dioxide levels. We're looking at how they mature. So over 2 years, scientists measured the survival rate of the sand lance embryos allowed to develop and mature at different temperatures and as well as different levels of CO2. So again, our null hypothesis is looking at the independent variable has no effect on the dependent variable, right? So in this case, our independent variable would be the temperatures that we're looking at or the carbon dioxide levels, which is all showing climate change. And what they're measuring, or our dependent variable, is the survival rate. So your options could be that the temperature will have no effect on the sand lance survival, sand lance development, or the size of populations. Okay, so again, the temperature has no effect on the sand lance survival rate. So they've given it to you all in that prompt. You just have to pull that information out. Okay. And the student, because I still have the student's answer in here, it says the changes in carbon dioxide and temperature levels will have no effect on the sand lance population and their survival rate. So other things we might see is a negative control and a positive control. Negative control is the absence of the treatment. So we have not given them whatever the treatment is. And so we're able to say the treatment does have an effect because we can compare our data to our control because it went through all the other parts of the experiment. It just didn't get the treatment group. A positive control has a known response. And it's a test of validity of the experiment. So if I'm looking at different solutions of, I don't know, runoff from plants, like these, power plants or whatever, okay? And I want to know, does it cause mutations? Then I can compare it with a sample of something that I know does cause mutations. And I can compare that, okay, here's how many mutations will be made from this chemical that forces mutations, and this is how many mutations I had that came from the power plant. And you can be able to then compare that because you know that that then creates validity. So they've given us this really long prompt. I really don't feel like reading this. You can pause it and read it yourself if you want to. But the things to focus on here is that group 1 contains no kinetic core proteins attached to a hotspot. Group 2 contains a CTF, and group 3 has an IML. Okay. So identify the control group. Well, since I'm looking at CTF and IML, I'm looking at the frequency of crossing over with them. My control is going to be the one that does not have that. So that would be this first group, or the one that is none, or the one that does not have the kinetic core protein. So group 1, the none, or cells with no kinetic core protein attached to their chromosomes. And the student also, I have their response. So we're now investigating whether red blood cells of animals lose ability to take in glucose of the environment as they age. So they incubated an equal number of red blood cells in separate culture dishes that contained a certain solution, and then they were testing those. So justify why they use the equal numbers in the dish as a control. So the whole point of a control is to allow you to be able to compare your data. So why would I want there to be equal numbers of cells? Well, if I'm looking at that, the age of the cell can have an effect on the amount of glucose that's being brought into the cell, then by having the same number of cells, I'm going to be able to compare the differences in that transport. Versus if I had different numbers, if I had more cells, I have no way to know, is it because there's more cells or is it because they had better diffusion transport? So we want to attribute the differences to the age of the guinea pig. We want to be able to compare these different dishes appropriately. We want to eliminate number of cells as a variable, and then the number of cells might kind of affect the results of them. Okay. So here we're given that same diagram where I didn't give you the prompt. Identify independent variable, which we already did. We want to identify a negative control in the experiment. So as I said, negative control will be the one that does not have the treatment group. So if I was treating it with GTP or treating it with the cholera, then I want to have the group that doesn't have either of those. So that would be sample 1. Now they also went a little extra step and they said, okay, well, if we were comparing how GTP had an effect, then my negative control would be looking at samples 1 and 2 because the cholera stayed the same in those. And I could then see, okay, what does GTP alone do? And then if I was looking at cholera, I could probably also look at how the GTP stays the same. And so that could be samples 1 and 3. So they actually gave you a lot of different options of how you could have put this. The logical answer would have been sample 1, and I believe that's what most students said, but there were additional answers based on what you were looking at as your independent variable. So you could have said single at 1, or you could have said 1 and 2. They also said just sample 2 as an option, or they said samples 1 and 3. So then they asked, okay, well, justify why the scientists included sample 3 as a control treatment in the experiment. So if I look at 3, I can see that it doesn't have GTP, but it has the cholera, okay? And so this would allow me to compare what the effect is, right? So I would be able to compare it with 4 to say, okay, well, this one has cholera, and it's going to be allowing me to be able to see, okay, well, what does GTP actually do for the cyclic AMP production? And as you see, it does significantly increase it if we have cholera and we either have the GTP or don't have the GTP. That has a significant difference. So calculations, we want to make sure that we show our work. And so a lot of y'all don't like to show work. So let me tell you why I say to show your work. So if you type everything into your calculator and then you just get an answer out and you give that answer, that's great, cool, whatever. But let's say you got it wrong. You typed something in wrong, you pressed the plus instead of a minus, like you messed up somehow, right? If you show your work, there's a way that like maybe the prompt, and this isn't always given, but maybe on the prompt it says like use the appropriate formula. So instead of your answer being right, use the appropriate formula and you showed your work, that might also provide you ability to get the points. So I recommend just showing your calculations just in case. You also want to not forget your units. So make sure you put the units on your calculation. And then the percent change has shown up a couple times on the exam. And this is now on the formula sheet. So let's just talk about it real fast. We have our final minus our initial, and then we're going to divide by our initial, and then we're going to multiply by 100. It's one of those science practices. You did this in lab. You probably did this with the diffusion lab. So they're saying that we are going to calculate the percent change in APA's activity in wild type yeast in a high P environment compared to a wild type and a low P. So we're going to look at these two numbers. Okay. So I'm comparing the high to the low. And so we're going to do our final minus our initial over our initial times 100, where our final is our 12.5, and then our initial is 0.5, and we'll divide that by 0.5, and then of course multiply by 100. So that's going to give me 3,360%. So just because you get a really large number doesn't mean you're wrong, okay? They did another one where they said, okay, we'll describe the effect of the cholera on the synthesis of cyclic AMP and calculate the percent change in the rate of cyclic AMP production due to the presence of cholera in the sample, okay? So we want to calculate that percent change, okay? So if we're looking at samples 1 and 3, we of course can, I think I'm ahead of myself. That was the describing. We don't want to look at that one. I want to look at percent change. So the percent change, I can say, okay, well, if I'm comparing it to 3, right, I'm going to have that I've got 127 subtracting 10 because my initial was 10, because without it to with it, right? So then I'm going to divide by 10, which gives me 1,170%. So again, the number is way high. So I hope that this was helpful. Remember that AP Bio Pain was just success. Bye, y'all.